All right. Good afternoon, Professor. It's take two. Just messed up on the last one. But this is our fourth Build a Lecture assignment. And on today's, I'm going to do Chapter 9. So let's get right into it. First thing we're going to look at is microbial nutrition. This is nutrients that are gained from the environment to help their cellular activities. There's three kinds. You got your essential nutrients, your macronutrients, and your trace elements. So the essential nutrients are any element or molecular matter that must be given to an organism. Our macronutrients is needed in somewhat bigger quantities, and it plays a vital role in their metabolism and their structure. And we got our trace elements, and usually that's seen in smaller quantities, and that plays a role in the function of enzymes and protein structure. All right, now we look at organic versus inorganic nutrients. So organic nutrients is all organic nutrients that must contain carbon and hydrogen atoms. Most organic things are the product of the living. Now let's look at the inorganic nutrients, which are molecules or atoms that contain atoms that are not hydrogen or carbon. So anything that has hydrogen and carbon are organic. Without them, they're inorganic. Now for our carbon sources, we got two types, autotroph, heterotroph. Autotrophs, they can feed themselves, and they can convert CO2 into compounds of carbon, and they do not need nutritional help from other organisms. Whereas heterotrophs, they need carbon from an organic form, and they also depend on nutrients from living organisms. They must have it. Another thing for heterotrophs, they usually are bigger molecules that need to be digested, enable for them to absorb them. Carbon sources live in a form that's simple enough for them to absorb. Now our nitrogen sources. The main source of nitrogen is in our atmosphere. That's the main reservoir for heterotrophs. They need to be broken down into basic building blocks and able to be used. NH3 is the only form of nitrogen that binds them with carbon to synthesize amino acids. Some bacteria and algae, they use inorganic nutrients like NO3 negative, NO2 negative, and NH3. In any circumstance, nitrogen has to be converted into NH3 before it goes into the gas. It also cannot reach structures of proteins, DNA, RNA, and ATP. All right, now we move on to some of our sources for oxygen and hydrogen. So oxygen sources, that plays a vital role in the structure, in the enzyme function, in the structure of the cell. 20% of our atmosphere is also made up of oxygen. Oxygen is a major component in our carbohydrates, our lipids, our proteins, and our nucleic acids. And it's common of inorganic salts. That's it for the oxygen. Now move on to the hydrogen. It replays roles in biochem of cells. So it maintains pH levels, forms hydrogen bonds with other molecules, and hydrogen is also free energy. So it's a free source of oxidation reduction. Okay, now we can look at phosphorus and sulfur sources. For our phosphorus sources, its primary inorganic origin is phosphate or PO3 negative. That is concluded made up from phosphoric acid or H3PO4. And that can also be found in oceanic minerals, rocks, sediments, things like that. The phosphorus sources, it's a key component of our nucleic acids. It's vital to the genetics of viruses and cells. And it's found in ATP, which is vital for our energy for the cells. Now for sulfur sources, that is distributed all over the environment, but it's mainly found in rocks and sediments. It's a vital piece of vitamin B1 and essential amino acids, methionine, and cytosine. It makes disulfide bonds that help shape and structure proteins. So let's look at organic nutrients and how the microbes feed off of them. One is the growth factor. which is organic combination of amino acids, nitrous bases, or vitamins that's impossible for them to be synthesized by any living organism. Those nutrients must be produced by the environment. So how do these microbes feed? There's two different nutritional types, which is phototroph and chemotroph. Phototrophs are microbes that photosynthesize, and chemotroph is microbes that get energy from different chemical compounds. Excuse me. So, two other types, photoautotrophs and chemoautotrophs. Photoautotrophs, they make the base for most of the food chains, and it makes vital organic compounds that are used by heterotrophs. Photoautotrophs also capture energy from the sun's light rays, and they use those rays to make chemical energy for cell metabolism. Now let's look at the chemoautotrophs. There's two main types, chemoorganic autotrophs and chemolithotrophs. They use organic molecules for energy and inorganic compounds as a source of carbon, and they depend on inorganic materials and does not need sunlight or organic nutrients. Okay, we look at our methogens. That is chemoorganic autotrophs that make methane using CO2 or hydrogen gas. And methane is a great fuel source and is used for greenhouse gases. Methogens, or some archaeas, they can live in extreme environments like ocean vents, hot springs, up to 400 degrees Celsius. They develop in anaerobic environments that contain hydrogen, like soil, mud, swamps, and even the inside of animals. Chemoheterotrophs. Chemoheterotrophs make up heterotrophic microorganisms. They determine carbon and energy from organic molecules and compounds, and organic molecules are treated by respiration or the fermentation that releases the energy in the form of ATP. Right, now let's look at aerobic respirations. It supports photosynthesis. Oxygen and glucose are reactants, and CO2 is produced. And the energy of Earth's balance is vital to the aerobic respiration system. Another thing, its principal energy yielding pathway in animals, protozoa, fungi, and even some bacteria. Okay. We can take a look at the I might butcher this one, saprobic microorganisms. It's mainly decomposers of animal matter, dead organisms, microbes, plant litter, all that good stuff. They contain a rigid cell wall, and they're not able to consume larger food particles, and usually they're bacteria or fungi. Let's look at our parasitic microorganisms. They can either live on or inside a host. They can cause harm and life-threatening or minimal. They can cause life-threatening or minimal harm. They are deemed pathogens because of the damage they do to our tissues. Another thing about these parasitic microorganisms, they are obligate parasites, which are unable to grow outside of a living host. And then there's obligate intracellular parasites, and that spends all of its time or part of its time inside a host. Okay, we can look at diffusion. Diffusion is the migration of compounds along a gradient from a place with a larger concentration or density compared to a lower density. Diffusion across the cell membrane is determined by the concentration gradient and the permeability of the substance. Now we can look at the movements of osmosis. Osmosis is when water moves beyond the selective permeable membrane. Osmosis is when the water moves beyond the selective permeable membrane allows. The differential membrane allows water to pass through, but not other dissolved molecules. We can look at the isotonic conditions. That's the cell's internal environment that has the same solute concentration as the external environment. It's usually isotonic solutions that are the most stable environments because they are already in an osmotic state. Water diffusion is at the same rate in both directions, back and forth. Go look at our hypotonic conditions. That is the external environment has a solute concentration that's lower than the internal environment. It has cells without a cell wall that can swell up and sometimes possibly burst. The most hypotonic environment is pure water because it passes no solutes. A hypotonic solution is the net direction of osmosis. Just did hypo. Now let's look at hypertonic conditions. The solute concentration outside the cell is a little higher than it is inside the cell. The cells use salt concentrations to preserve its food. It limits the growth of microbes, and the high osmotic pressure forces water to diffuse outside of the cell. Now we can look at hypotonic and isotonic adaptations. Isotonic conditions only put a small amount of stress on the cell. Moving on to hypotonic, their environments include amoeba and bacteria that thrive in freshwater ponds. Bacteria possesses a cell wall that prevents the cell from rupturing. And amoebas contain a vacuole that removes all the excess water in the cell. They also have a high vital need for higher salt concentrations. Now we can look at hypertonic. Scratch what I said on that last one. On the hypotonic, that limits the growth of microbes, and that has a higher pressure forces for water to diffuse outside of the cell. Okay, hypertonic adaptations. These cells have to choose between increasing the salinity within the environment or restrict the loss of water. Halobacteria is a type of hypertonic cell that absorbs the salt to make the cell isotonic. These hypertonic adaptations have a vital need for higher salt conditions. That's what I was missing. That's it for me today, Dr. Dworsky. I'm glad I got this one in. And yeah, have a great day. Talk to you later.